Good morning. Welcome you all to the next class on inorganic chemistry of life, principles and perspectives. Let's look back into what we have done in the previous class. Just in the previous class, I have started regarding the zinc enzymes and explained to you there are at least 200 zinc enzymes functioning in human body, having more than half a dozen different kinds of functions, and I have brought the different types of functions to notice, and some of these you can see once again here: hydrolases, peptidases, lyases, then you have oxidoreductase, transferase and ligase. I mentioned to you, in spite that the zinc does not undergo redox, zinc containing enzymes have got activity of the oxidoreductases. I have also explained to you in the previous class that the cofactor is involved in doing such kind of a redox mechanism, and this has been explained very well by the example of a example that is using the alcohol dehydrogenase. Okay, alcohol dehydrogenase is the enzyme which takes this alcohol to aldehyde, and also it is reversible kind of thing from aldehyde to alcohol. But instead of zinc undergoing redox, it is the NADH NAD plus is the couple. So, as you can see here, NADH NAD plus, these are the things that happens. So, instead of the zinc undergoing redox and the cofactor, additional cofactor which is NAD plus NADH. So, that means in the zinc vicinity, the NADH NAD plus should be poised in such a way that the reaction triggers, and that is what I explained to you in the previous class. Also, I explained to you there are zinc centers which having one zinc metal ion. There are zinc centers which are having zinc enzymes which are having 2 or more. For example, here you can see that there are 2 zinc, there are 3 zinc, there are 3 zinc, etc. So, we have a range of zinc containing enzymes. So, taking you once again through the alcohol dehydrogenase, that you can see the aldehyde to alcohol, alcohol to aldehyde. So, oxidase form requires to reduce this one, NADH goes to NAD plus. So, and then the other way is NAD plus goes to the NAD. So, I have explained that the mechanism where the cofactor lies in close proximity to the zinc center, and zinc center does not undergo redox, but the redox will be happening at NAD plus NADH, as shown here the way that the orientation of this. I have also explained to you there are 2 subunits, the large and the smaller one, where you have the, of course in this case you have 2 zinc ions, one zinc center is catalytic which you see here, other zinc center is with the tetra cystenyl one, which is a structural one. Also you can see the coenzyme binding domain, and then you have your substrate entering into the thing. So, these are all we have looked at, and we gone through all this mechanism. So, let us look at with a newer enzyme, and this is an enzyme which is nothing but hydration of carbon dioxide. You know, in the tissues, body cells, tissues, etc., you always have certain CO2 being present there. So, in order to maintain acid base balance in the body, in the tissue, in the blood, so one needs to sort of remove the CO2 by bicarbonate, and the vice versa. So, therefore, you can clear the tissues from all these species. So, the enzyme which does is the carbonic anhydrase. So, if you take the carbonic anhydrase, it requires a zinc ion, it is a must. Without the zinc, it does not show. If you take the carbon dioxide in water, you would see the conversion, but very slow. But when you put this enzyme, the reaction goes to 10 power 4 to 10 power 6 per second. So, which is the reaction is expedited by this enzyme. That means, enzyme is involved in converting the CO2 to H2CO3. So, this is how the acid base balance is being maintained in the body. In lungs and in nephrons and the kidney, etc., low CO2 concentration, and in plant cells also. So, in all the things the CO2 needs to be balanced by this. Okay, let us look at the spectral properties of the carbonic anhydrase. Can we get good spectral properties of carbonic anhydrase when zinc is present? No, because what is the reason? The reason is zinc is in zinc 2 plus, zinc 2 plus is D10, D10 does not give much of a magnetic kind of thing, spectra. So, what you need? You need to replace this by the cobalt. So, initially you take the carbonic anhydrase, you remove the zinc by using certain kind of a chelator, and then you can create a apo enzyme. So, this one can be titrated with cobalt. So, you can get cobalt embedded. So, you can see the protein over there, and this is the zinc position. This position, the zinc can be removed and replaced by the cobalt, so that you have a cobalt containing carbonic anhydrase. Now, on this side you have a number of spectra, as you can see, whose intensities are varying, and these spectra have come because of what? By varying the pH of a cobalt containing carbonic anhydrase. So, we take the cobalt containing carbonic anhydrase and look at the absorption spectra as a function of pH. And you see something like the spectra, the intensity is going up, the intensity is going down. As a result of this, you have a crossing here, you have a crossing here. So, what is this crossing known as? Isomestic point. So, when do one find such isomestic point? When there is a kind of a transition occurring from one species to other, or when there is a binding of a metal ion or dissociation of that. So, events that are taking place. So, that means there is a real interactions going on. So, now if you look at this particular band as a function of pH and try to plot its intensity or absorbance, either way is okay, epsilon or intensity as a function of pH, you see that the curve that is going. So, this one is the one where you have more than one sigmoidal. That means there are more than one possible ionization, deionizations are possible, and this is, as you can see, there are two ionizable groups, the water and the protein side chain, and that is why you are finding one here and then one more here. So, therefore, from this pH titration you can get definitely the pKa values and the information as I mentioned earlier. So, what is the reaction? We know very well, CO2 plus H2O, HCO3 minus plus H plus, and this reaction is if there is no catalyst, that means no enzyme, the reaction is 10 power minus 11 per second, and if there is a catalyst or an enzyme, it is 10 power 4. See the kind of expedition. So, what is the real rate of enhancement? Real rate enhancement is from minus 11 to plus 4, which is nothing but 15, 10 to the power of 15. So, that is a huge level of increase in the reaction rate. Okay. So, as you can see that the zinc with the 3 histidines and the water, and this is being sort of coupled by a side chains of threonine, etc., and glutamate in a sequential way. Now, let us look at the details of the reaction mechanism of this. For the details of the reaction mechanism of this, so you have a zinc center, as we have seen already, and you have a protein here. So, you take this curved line kind of thing as a protein. You have certain groups which are protruding here, for example, threonine, and this is being coupled by a glutamate. So, the glutamate acts as a general base. So, this base can pull out the proton over here, and that can trigger the reaction over there, and that leads to the further reaction to take place. Now, in a this resting enzyme, what you have a water which is connected to this NH by a hydrogen bond, and which is also forming a hydrogen bond with this zinc hydroxy. Now, when the CO2 comes in, actually there is a conformational change comes in and it will knock out the water out and binds, takes the position of the water and thereby binds in this way. Now, what is this step is called the recognition. So, in this fashion the CO2 is recognized by the enzyme through this kind of a hydrogen bond, and this enters in as a result of replacement of the water, that brings appropriate conformational change, and this is the things that you look at here. And now, as a result of the replacement of the water and attaching the CO2 through a hydrogen bond, will place the carbon dioxide in strategically in such a way, it is placed in a strategic manner such that the zinc hydroxide oxygen can have a nucleophilic attack. Let us look at for a while how you get the zinc hydroxide. So, if initially you have a water and the water is being activated by this group, neighboring group, and that basically it was the glutamate which will remove and make the hydroxide. And you know that the hydroxide is a base, and if when it bound to the zinc center, will it be the equivalent as strong base as OH minus? You take a sodium hydroxide, put in water, you know it is a very strong base. Now, you put the OH on top of zinc 2 plus, which one is a stronger base? So, the stronger base is free hydroxide, not zinc hydroxide, but however this enzyme prefers zinc hydroxide, not the free. Why the reason? Any idea about this? Reason is that because this hydroxide whichever is formed has to have an attack or reaction or nucleophilic reactivity at the carbon center of the CO2 and nowhere else. On the other hand, if you have a free OH, the free OH can start deprotonating many groups in the protein. So, therefore, free OH is not favored by the enzyme, and the zinc bound hydroxide is, because this cannot go and take out any proton that is required because it is far away. So, it can only give a nucleophilic attack on the carbon center of the CO2 because CO2 is bound in such a strategic manner at this position through a hydrogen bond. And this is what is an important aspect of it. Now, this whole thing you can call it as a CO2 recognition by enzyme. Now, once you have the CO2 placed in a proximity to the zinc center, proximity to the zinc hydroxide will take the advantage of having a nucleophilic attack, and that nucleophilic attack will give basically a kind of a intermediate like this. So, this here and this minus, it will go to plus. So, you will get some kind of a metal loss cycle. So, this is the carboxylic group with a metal loss cycle, and this is the basically an intermediate. So, this intermediate is broken in presence of the water, and you can see that, and that will leave out the HCO3 minus, and then this upon the protonation will give the next step back to the normal one. So, therefore, enzyme catalysis is complete. So, enzyme catalysis is complete. So, simply you can say, so zinc 3 histidines in water, and this is replaced by some general base from the protein's side chain will knock out the proton and create the zinc hydroxide. Zinc hydroxide will attack on the CO2 not in a free system, but in an enzyme system, because it binds to that, and that will give an intermediate, and this intermediate upon hydration will give HCO3 minus. So, what is the role of the protein? Role of the protein is to provide the position recognition for the CO2 and the position with respect to zinc hydroxide, so that the zinc hydroxide can have a mechanism of nucleophilic attack on this. Hope you understand now this whole system. In enzymes, always enzyme has to notice, has to identify, has to recognize the substrate. Here the substrate is CO2, the CO2 is recognized through the hydrogen bonding interactions of this, okay, and places in such a way that this is close by, and the rest of the thing is quite well understood on this. Okay. Now, so that takes care of the how carbon dioxide is converted the H2CO3 etc., and then maintain or balance the acid base balancing in the tissues by the carbonic anhydrase. Let us look at another enzyme. We know very well a large number of peptidases are there which are based on zinc, and they are all involved in the breaking the protein into individual either peptides, smaller peptides, or even amino acids, and that is how you get the amino acid pool to synthesize the protein. Okay. So, in that you have peptidases which are carboxypeptidase, also there are peptidases which are aminopeptidase. What is this nomenclature from? Carboxypeptidase means the enzyme is active in the C terminal or carboxylic terminal of the protein. This is active in the C terminal of the protein. And aminopeptidase, in the aminopeptidase it is basically active in the amine terminal of this. But in this particular course we will be studying only carboxypeptidase, not the amine peptidase. So, now you got one thing. So, what we understand? We understand that the carboxypeptidase can cleave the peptide bonds at the C terminal. What kind? What kind is dependent on the type of the enzyme. If you have a neighbor which is aromatic, if you have a neighbor which is cationic, if you have a neighbor amino acid which is anionic, there are different kinds of enzymes. Carboxypeptidase A, where the neighbor amino acid is the aromatic branch, and if you have a positively charged amino acids like lysine, arginine etc. in the neighbor amino acid to the peptide with respect to the peptide which you are cleaving, and then this will be carboxypeptidase B, and if you have a negatively charged kind of a species in the neighbor, then it will be glutamate carboxypeptidase. This is also called peptidase, carboxypeptidase C. Carboxypeptidase A, carboxypeptidase B, carboxypeptidase C. Let me explain you a little more understandable way. Now you have you know that you have a carboxylic terminal or an amine terminal, then you have a C alpha, and then you have CO, and then you have NH, then you have C alpha, and then you have COOH. Okay. So, you are looking at the peptide. Now, you need to look at this R1, you need to look at this R2. So, therefore, if you have at the end this, if you have this as aromatic etc., then you then this will be carboxypeptidase A, carboxypeptidase. So, this is we refer it as CA, CP A, and if you have the cationic residue as a neighbor, then this is CPB, and if you have a anionic residue, then it is the CPC. So, carboxypeptidase which can cleave C terminal, if there is aromatic it is called carboxypeptidase A, and if you have a cationic residue at the amino terminal carboxyl terminal, then it will be carboxypeptidase B, and if it can cleave in a peptide bond where you have a anionic residue towards in the neighbor of this particular peptide bond. So, this then it is carboxypeptidase C. So, basically what you are doing is you are hydrolyzing this. So, you are adding water here, that is what is the peptidase. So, what you will get? You will get the corresponding COH, so corresponding NH2, that is what you will get. So, now you understand there are 3 different types of the peptidases: carboxypeptidase A, carboxypeptidase B, carboxypeptidase C. All of these will hydrolyze the peptide bonds in the carboxylic terminal, but depending upon what the neighbor residues are. If the neighbor residues are aromatic or branched aliphatic, then carboxypeptidase BA. If you have a positively charged species as a side chains in the neighbor, then it is carboxypeptidase B. On the other hand, if you have an anionic kind of a species at the carboxylic terminal, then it will be carboxypeptidase C. Now, having got that, let us look at the enzyme core. So, this is the enzyme and this is the zinc ion, this bound. So, if you expand this region, you can see this is the zinc ion, there is one carboxylic glutamic carboxylic group. So, number is not very important, it is center in this glutamate 272, it binds like a bidentate, and there is one histidine over here, there is one more histidine over here, and there is a water molecule. And there are some additional residues far away, and this will be you can refer it as a secondary coordination sphere. These will form the secondary coordination sphere. They are helping the process of primary coordination sphere, whatever happens in this. What is the reaction? As I have shown you on the earlier by writing that you have the peptide bond, you add a water, then you will get a carboxylate, you will get amine. That is what basically reaction is, and this is what you refer to as a cleavage, peptidase. In other words, you can also say hydration of peptide bond. Hydration of peptide bond is same as peptidase in this case. Now, you can see in one of the case, probably is the carboxypeptidase kind of thing, then a glycyl tyrosine here. So, this is a glycyl tyrosine. So, this is glycyl tyrosine. So, this is NH2C alpha. So, since it is glycine, it will be CH, and CO, then NH, and what you have C alpha. Here what you have is if it is a tyrosinyl, you will have OH, and you will have the next group. So, which is the CO2 minus. So, this is the amine terminal, this is the carboxyl terminal, this is the glycyl tyrosine. Now, you can see now in this particular the slide, and this is the center where the zinc is bound to the one glutamate and two histidine with the water, and this the peptide when you bind, of course, it can bind over here, and this has been checked by crystal structures, and this is actually poised because this particular tyrosine, when it binds, there is a conformational change. You see the tyrosine which is looking outside in a resting enzyme. When the substrate comes in, this is the substrate, and this turns into this side. That means there is a purpose. So, how do you know? You look at the simple resting protein enzyme structure and the enzyme structure in presence of the substrate. Can you ever crystallize the presence of the substrate? No. So, you use something very mimicking to that of the substrate, but it will act as an inhibitor. So, inhibitors can be easily crystallized and studied, and that is what is studied over here. So, that is because this hydroxyl group is in close proximity to that particular peptide moiety. That means this is basically required. Okay. And another way of understanding this is that the zinc with the water can be converted into easily by the glutamate neighboring here into hydroxide. This hydroxide can attack at the peptide bond and thereby form a kind of a intermediate, which is the 4 membered intermediate, just like the one which we have seen in the previous case for the hydration of carbon dioxide. Okay, so therefore, the carboxypeptidase, as I said in the previous case, which is the case of the carbonic anhydrase, always there should be a recognition, the right substrate should be recognized, and then there should be a reaction occurring. So, therefore, in this case, let us break the steps into the form of 3 possible steps. One is substrate recognition. So, then next is a peptide bond cleavage, and that is not sufficient. Then the product has to release, otherwise still bound. So, the whole reaction can be treated in terms of the 3 possible steps. A step of substrate binding recognition, a step of cleaving the peptide bond, a step of releasing the product. Now look at the A and the B and the C here. So the A, as you can see here very nicely, this is the kind of a thing where this is a different dipeptide, which is the real dipeptide on which the enzyme works, and this is nothing but your so this is nothing but your NH glycyl, glycyl. So this is the glycyl, this is the peptide bond, and this is the C alpha. Here you have CH2 and here you have phenyl. So and then you have CO2 minus. So this is the carboxyl terminal, it will recognize from this, and this is blocked by some other group here and okay. Now this is the peptide which is cleaved. So this is nothing but benzyl, NH is coming here, glycyl, and this is phenylalanine. So this is the peptide which you are hydrolyzing. So it is this COH bond which you are cleaving here. So this is where you add water or cleave. So this is the real reaction on enzyme, and that is what is seen in this particular thing. So you can see that this is when this binds, this is being recognized by the CO2, the neighbor residues. There are 3 hydrogen bonds. So the arginine and the other groups, the tyrosine and asparagine, all these kind of groups will make and this is reason why this is being poised in very nice way close to the zinc hydroxide. Just like the previous case, the CO2 is poised and here the peptide moiety is poised. So therefore, and this is activated by this glutamate, and this glutamate will make the hydroxide, and then either you can have a zinc bound water which is activated by glutamate, or a simple water which is activated by glutamate, but finally the attack is on the carbonyl of the peptide, and this is a nucleophilic attack. So this is how it recognizes. It not only recognizes the C terminal, it will also recognize the N terminal by hydrogen bonding. So the entire dipeptide sequence is recognized. So that is why it is called carboxypeptidase. The carboxyl terminal of the peptide is recognized. So that means last 2 residues are being recognized. I hope you understand that. So all these small the dashed bonds are recognition basically. Now this one, once you have this is activated, this water is activated or zinc hydroxide is created, that will create a active bond through a nucleophilic attack here, and that will give the intermediate, as you can see here. So this is actually a step towards the breaking of the CO bond, the CONH bond, and here you can see the CONH is bonded, is broken, but still the thing is connected. Therefore, this connected thing can be basically taken out by adding water. So that will take out the carboxylic part of the thing, amino part of the thing as 2 products. So you can see the step, the recognition, the nucleophilic attack, and the actual cleavage, and the release. The release is by adding the water. So this whole thing, whatever I mentioned, can be seen in a little different way. This is nothing but the center, the zinc with the primary coordinate sphere and the secondary coordination. This is one of the secondary coordinations. So these secondary coordinated groups are either useful in recognizing the substrate, which is a dipeptide, and or both. Also it is involved for some protonation, deprotonation kind of steps. Now let us start from here. This is your free enzyme, a rustic enzyme, and this is water is activated by the glutamate, and then hydroxide is formed. This can be talked either the zinc bound water or the free water, it does not matter, and that will attack on this peptide, forms an intermediate which is the 4-membered intermediate, and this further undergoes the rearrangement and then breaks this particular bond, and then here when you add the water and the carboxyl terminal and the amino terminal will separate and then go back to this. So, I hope you understand the kind of a mechanism. So, in this particular class what you have seen two things. One is addition of the water to CO2 forming the H2CO3 or carbonate kind of a species. In the second case, C-terminal carboxypeptidase, where two of the last two residues are being recognized. Which are the last two? Carboxy terminal of the last two are recognized, and these are acted by the water converted into hydroxide or zinc bound water converted into hydroxide, which gives a nucleophilic attack to result in a four membered intermediate, which in turn rearranges to break this as a result of the nucleophilic attack, and in presence of the water this will dissociate to carboxyl terminal and the amino terminal groups of this. So, that is the kind of thing. In the next class we will look at other kinds of enzymes of the zinc. Thank you very much.